I'd like to talk a little bit about this webinar before we get started. This webinar is part of an exciting series created by the Plant Gene Steering Committee. It's called the Interactive Series for Plant Transformation Protocols. And this is not a traditional workshop, it is an interactive session. So we'll be taking time throughout the event for audience questions. When you do have a question, please put it in the Q&A box, not the chat. So the Q&A box helps us manage the questions. So again, please put your questions in the Q&A box. I'd like to briefly introduce today's speakers. We have Kiflom Aragawi. He is a staff research associate at the University of California, Berkeley. He received his master's degree from Wageningen University in research. Sorry, I probably slaughtered that name, but it's in Belgium, I
No,
believe, right?
Netherlands.
Oh, Netherlands. Okay. In the Netherlands. His research focuses on developing transformation and genome editing tools for cereal crops, with particular emphasis on sorghum and soteria. Kiflom also has experience in gene function studies and in improving sorghum traits through genome engineering. His current research focuses on CO2 assimilation and sequestration through genome engineering. Jianchen Shen is an assistant project scientist in the Department of Plant and Microbial Biology at UC Berkeley. His interest in plant science began in Wuhan, where he earned his bachelor's degree in biological sciences from Hiaozan Agricultural University. Motivated by a strong interest in gene regulation, he pursued a PhD focused on translational regulation responding to environmental stress in crops. Following his doctoral training, he conducted postdoctoral research at the University of Pennsylvania, where he studied epigenetic regulation. He later transitioned to UC Berkeley to focus on sorghum, genetic transformation, and genome editing. Since 2023, he has held his current position where his research is developing high-efficiency genome editing strategies for engineering sorghum for soil organic carbon sequestration. So thank you again to our speakers for taking the time to be here with us and for sharing their expertise with us. So hopefully you guys all read the protocol before coming here today. If you haven't, I'll put the link in the chat shortly. The protocol is divided into two sections. Jian Shen is going to go through the first few slides. We'll take a break, do audience questions, and then Kiflom will do the second part. Again, if you have questions, please put them in the Q&A box. As you're thinking of them, I'll be moderating questions for the speakers. And just a reminder, any information presented today will be available on the PlantGene website, along with a recording. All right, thank you, and I'll turn it over to our speakers.
Good. Okay. Thank you, Amy, for warm intro. We will start our workshop today. My name is Jian Chang, and I've been here for sorghum transformation tool development. And let's start our protocol. So first slides just want to show you the brief idea of how we do sorghum transformation. And we use the immature embryo strategy. Basically, is to isolate the 1 to 2 millimeter immature embryo and then infected by the agro. So it would take about 1 week of co-cultivation, 1 week of resting, and 4 to 8 weeks. Sometimes 6 weeks is sufficient, but we will extend it to 8 weeks regeneration and 2 weeks for rooting. So in total, it's about 3 months from the agro infection to the point we get the regenerants. And also, here is what we have in terms of the transformation frequency. With the baby moon wushou, and the transformation frequency is more than 50%. As with wushou only, it's about 20 to 30%. The next slide. So today I will mainly focus on the first part, which is the immature embryo isolation and agro preparation. Yeah, so we will start with agro. So agro take about 3 days to grow. So then here we showed it's about 3 days before the transformation, and prepare the mass mix. So it basically is to strike the agar strain. In our case, is LBA 4404 Steinmeier strain on a YEP media. YEP is a yeast extract peptone media, and then keep at 28 Celsius degree for 2 days. 1 day before transformation, we have to prepare the working plate. Is basically to pick 5 to 6 colonies and strike on working plate. It's also the same YEP media and keep them overnight, 16 hours. So here is what the hmm, plates look like, and from working plates we will get some agar culture and to prepare agar suspension. Yeah. So the next part I will start from panicle. So here the panicle should look like, and the aim of this step is to obtain about 100 to 200 immature embryo. The size would be 1 to 2 millimeters. So on the day of the transformation, we took about 2-week-old post-anesthesis panicles. So here is the panicle look like, and we will take the middle part. And here the seeds look like it's about the sepal or the glume cover half of the seeds, and then we isolated them from the panicle and keep them in the 15 ml Falcon tube. With that, we will remove the glume and keep them, trying to keep them intact. Otherwise, the surface sterilize might like disturb the immature embryo intact. And this is a surface sterilization step. So we use 75% of ethanol and rinse them twice, each time for 2 minutes, as we shared in the protocol. And also we will rinse by water for like 2 to 3 times. And after that, we will treat it with 20% bleach with 0.1% twin 20. After we finish all of them, we will keep them on petri dish, basically with a filter paper, and to air dry for 30 minutes. So this step is typical the time we can like stop or pause for a while, and then we will continue working on the immature embryo isolation. So the next step, I will show with you the isolation steps, and typically we will use the back of the plane and to press it, and then another hand we use forceps to hold the immature embryo and then move them to the infection media. But the key here is to keep intact IE. Otherwise, it's very hard to get it transformed. So I will show you the details by a video and press it. So we can see, and we just gently press the seeds and then move the seeds to the image to the agro infection media. So the speed wise, it's about 3 to 6 immature embryo per minute on average for me, and it probably would take an hour to isolate 100 seeds. So just keep your pace. And some if you are well trained, and the speed might be a little bit faster, but it's the same. Yeah. Okay. So and the next step is trying to prepare agriculture. So here I show is to how much agar we will pick, and it's about the right seed size, and then we will put them into the agro infection media. We will repeat this for 2 to 3 times until we get enough agriculture. So after that, we will adjust the agro concentration at OD 550. So the ideal situation to adjust them to 0.7, and then adding Silvet as well as the Cimetine. So Cimetine is a little bit unique for our strain because it's a supplement to maintain agar growth, particularly in co-cultivation stage. And here is the last step I would like to share. So after we isolated all the immature embryo, we put it into 2 ml centrifuge tubes and replace the media by agar suspension. After that, we will invert it gently for 5 minutes and then discard the agar culture by pipetting up. But the key point here is trying to remove all the agar suspension. Otherwise, it would have agar overgrowth issue in the next step. With that, I think I'm happy to take all the questions if you have any. Yeah.
All right, thank you. So we have some questions coming in. This is from Teal Row. Do you invert the immature embryos in infection constantly for 5 minutes?
Uh, yes. So you are talking about the agro infection step, right?
I believe so, but Teal, if you could just yes.
Okay, yes. So it typically take 5 minutes. So 5 minutes is sufficient for agro infection, and but it will keep longer. At Kiflin will share with us the co-cultivation stage. It plays on media for 1 week.
Okay, thank you.
Sure. Any other questions?
Yes, this question is from Mary. What's the efficiency or the efficacy of transformation? How do you calculate the efficiency?
Okay, that's a great question. So, the way we calculate the transformation efficiency is by getting how many. Okay, so let's take example. We will start with 100 immature embryo. As long as we get about 30 independent events from all the immature embryo we isolated, and we will calculate it as 30% is 30 over 100. It's independent events over immature embryo we start with.
Okay, thank you. We have another question from Chin Sen. How do the immature seeds need to be, or why do the immature seeds need to be dry before isolating the embryos?
That's also a great question. So, there are two things we need to consider. One is typically the surface sterilization and the seed harvest step would happen in the morning, and we probably would take some time off for lunch. This is the main issue, and for me. And another thing is if it's wet, it might increase the risk of contamination.
Okay, great. Um, we have this is kind of questions from two people that I'm going to combine. So we had a
Hmm.
question about the OD reading.
Hmm.
So if the OD reading at 550 is zero point 0.7, do you know how much it is for OD 600?
Mm,
Yeah, sorry. And then the second question to that is, do you grow the agro to an OD of zero point 0.7 or to a higher density and then adjust to zero point 0.7 by dilution?
okay, I will answer the second question, and we will dilute the agro suspension to 0.7 suspension. And for the first question, I be honest, I don't know because we typically use 550, and I don't know why. This is our experience. Kifin and I had a conversation about this, and this is just what we do. Yeah, and I don't know how much that was in OD 600.
Okay, thanks. And then, sorry, I think I missed this, but did you talk about growing it to a higher density and then adjusting by dilution?
Yes.
Okay, okay, great. I just want to make sure that answer.
It's okay. First of all, it's not growing. It just basically grow on plates, and then we take as like two to three scoops and put it in the liquid, make it a suspension, and then we adjust the concentration to 0.7 OD 550. Is that clear?
Okay. Um, Tim followed up with that question. Do you grow the agro to saturation?
Mm, this is a good question. We don't know because we are using the plates. So as long as we see enough agriculture for use, and we will start the transformation. Typically, it would take about 16 hours on working plate. And another reason, okay, I just wanted to add up. Another reason we didn't use the liquid culture. I mean, I know a lot of people they do agro uh, growth in liquid, but this for this particular strain, it would create or generate a lot of chunks if it's in liquid. So that's why we thought the solid plate would be more ideal for its growth. Yeah.
Okay, great. So this question is from Sarah. Can you explain why you add the Silhouette L77 and the thymidine to the infection media?
Okay, that's a great question. Silvet is to increase the infection efficiency and just break the part of the waxy layer at the top of the immature embryo, and the thymidine is a supplement because we are using the LBA 4404 thymidine minus, which means it's extrophic. Without thymidine, it cannot grow. So the reason why we wanted to use this strain particularly is because it's extrophic. It can prevent overgrowth on co-cultivation media. And the reason why we are precautious about the overgrowth is just because uh, immature embryo, particularly in sorghum, is relatively sensitive to stress. As long as it's overwhelmed by the agro infection, it will die.
All right, thank you.
Mm-hmm.
So I think you answered this, but the if you're growing aggro on a plate, how are you taking the OD and how are you determining it? So I think you mentioned this that you're culturing the agrobacterium, um, but I guess
Yeah.
that just needs some clarification.
Yeah, I need this need a clarification. So we suspend agar, so as we showed here, I can show you again. So after we grow agar on plates, and uh we will like take 2 to 3 scoop and into the liquid culture, basically the agar infection media, and then suspend it. And we measure the concentration in the agar suspension. Yeah.
Okay, great.
Oops. Is there any more questions?
So I think we are ready to move on to our next section. If you have questions about this section, you can continue to ask. And we will have some time at the end to talk about the whole protocol. So we'll try to get to as many of those questions as we can. All right, with that, Kiflom, I'll turn it over to you.
Thank you, Amy. I'll share my screen. So hi, everyone. I'm Kutlo. I will continue from where Jinjin stopped. As Jinjin mentioned, there are about 6 steps in sorghum morphogene-assisted transformation strategy. It starts from isolating the immature embryos up to transferring the regenerated plantlets into the rooting medium. After agro-infection, the next step is to remove the agrobacterium suspension culture from the 1.5 ml tubes and transfer those immature embryos into a co-cultivation medium. Even though we are removing the agrobacterium, still there will be some agrobacterium in contact with the immature embryos. That means the agro-infection process will continue during the co-cultivation period. Uh, when we culture the immature embryos into the co-cultivation media, we have to make sure that the scutellum side that you see on figure E is facing upward, and the flat size is in contact with the medium. If we do it the other way, normally somatic embryos are formed on the scutellum, and then if we invert it upside down, the immature embryos will not be able to form the somatic embryos. Afterwards, we can seal our plates with parafilm and incubate them at 24 degrees centigrade in the dark for 7 days. One thing that we have to pay attention at this stage for Texas 430, which is a common sorghum variety that people use for transformation, you don't see any phenolics or stress during this period, the 7 days period. But for some varieties, for example, 623 or some other recalcitrant varieties, you might see phenolics during this time. In that case, it's recommended to refresh the tissues into a new co-cultivation medium, even within the 7 days. So after keeping the immature embryos for 7 days on co-cultivation medium, the next step is moving those immature embryos into the resting medium to stop the agrobacterium growth. In our case, as Jinchi mentioned it earlier, we are using the autotrophic agrobacterium strain, LBA4404 Thi-, which can't produce its own thymidine. So to stop the growth of agrobacterium at this stage, we can remove thymidine from the resting medium. Additionally, it might also be important to add antibiotics at this stage to completely stop the growth of the agrobacterium. Tissues will stay in this medium for about 10 days. And during that time, we will start observing formation of somatic embryos, as you can see it in this figure. And if you include a visual marker gene in your construct, you will be able to see its expression under fluorescence microscopy. Especially if you are optimizing your transformation strategy, we recommend including a visual marker gene, so you will have an idea of how your transformation is doing at this early stage. In our case, just before moving the tissues into the next step, we need to do heat shock treatment to remove the morphogenic genes. As you can see it in here, the somatic embryos are already formed, so we no longer need the developmental genes Baby and Wushel. And if we continuously express these morphogenic genes, especially at a later developmental stage in the plant, there will be abnormal phenotypes. So to excise these morphogenic genes, we use a Krelox system. This is part of our construct that we use. As you can see it in here, there is Cre over here, and then there are 2 LOX-P sites. And the promoter driving the Cre recombinase is a heat shock promoter. So to initiate the Cre recombinase, we need to treat tissues at a higher temperature, around 45 degrees centigrade and 70% relative humidity for about 2 hours. This is an efficient strategy. It worked well for us. We were able to obtain an excision efficiency of between 75 to 85%. That means 75 up to 85% of the transgenic plants will not have the morphogenic genes. After heat shock treatment, the next step is to move the tissues into embryo maturation medium, which contains a selection agent. In our case, we use either imazepyr or hygromycin. At this stage, the somatic embryos, as you can see it on this figure, will mature. The tissue will proliferate, increase in size, and we will start observing formation of tiny plantlet. We will keep this tissue in this medium until we get enough number of plantlets that we are interested in. But of course, we have to refresh the tissue every week or every other week into a new embryo maturation medium. As you can see it also on this figure, the tissues are increasing in size. Through time, they will be larger and larger. We have to reduce the number of tissues that we keep in each plate to make sure that the tissues are having enough nutrient. At a later stage, when we keep refreshing these tissues into a new embryo maturation medium, and then at a later stage, we will start obtaining plantlets, which are about 1 to 2 centimeters in height. You can see it on this figure over here. And these plantlets are big enough to be moved into a rooting medium to initiate root development and also for further growth. Unlike the classical sorghum transformation strategy, people who do sorghum transformation probably are aware of, here we can get, using the morphogene-assisted transformation strategy, we can get multiple plantlets from each immature embryo. Of course, not all the immature embryos are going to give us plantlets, but the ones which survive on the embryo maturation medium most of the time give multiple plantlets, and we have to separate, if possible, each plantlet by itself, as you can see it here. It's important. This is important because especially when we isolate tissues from the plant, when we take leaf samples from the tissues at a later stage, and when we do genotyping, having a single plant will make it very easier. If you feel you are going to damage the plantlets, when you isolate an individual plantlet, it's okay to move 2 or 3 plantlets together into the rooting medium. But at a later stage, when they grow on the rooting medium, you might have to discard the extra planters. So you can create a little opening in the rooting medium using your tweezer and transfer the plantlets into the hole that you created. In general, I recommend transferring around 4 or 5 plantlets to make sure that the plants have enough space. And then at a later stage, if you included a lot of plants in one plate, it will be difficult. When you move these plants into soil, it will be difficult to separate the roots. Then these plants can be moved into a growth room with a lower light level with the following conditions to initiate a root formation. Within 2 or 3 weeks, most of the plants, they should be able to give well-developed root system and shoots, as you can see it here. If the plants are big enough, at this stage, I like taking leaf samples, DNA, extracting DNA from them, and doing genotyping. So I will transfer to soil only the plants which are positive for my target gene. It depends on the work that you are working. So once the plants developed a strong root and shoot system, they can be moved into the soil. We have to make sure when we move them into the soil, we have to make sure all medias are removed from the root, and we have to keep 1 plant per each pot. As soon as we moved it into the growth chamber, we have to make sure to cover the plantlets using a dome until the plant is acclimatized into the growth chamber environment. Normally what I do is when the plants reach the height of the dome can be removed at that stage. Finally, when the plants reach around 4 to 5 leaf stage, they can be transferred into larger pots in the greenhouse for further growth and generation advancement. So this is the last step. With that, we would like to thank members of the Lemo Lab, Peggy for her guidance, for her support, a previous member of the Lemo Lab, Judith, who started this transformation a while ago. We would also like to thank Jinchens and my undergrad students for their amazing work. Finally, we would also like to thank Bill for the constructors, for the helpful discussion we had, and Ping Chi from Corteva as well. So with that, I will take questions, Amy.
All right, so we have a lot of questions coming in. Just a reminder to everyone to please ask questions in the Q&A box, not the chat. You can find the Q&A box at the bottom of your screen. It is a little box with a question mark in it. So we'll start with the questions. This is from Golem. Why do you need to keep the sculetum side up? What are the benefits? Sorry, I probably mispronounced that, but
No, yeah, that's perfect. That's right.
okay.
Yeah. So the immature embryos, they only form on the scutellum side, like the upper side of the immature embryo, which is curved. So if we do it the other way, there's no way like those sides will be in contact with the medium, and there will not be like somatic embryos formation on the scutellum side. So generally, sometimes that happens by mistake because you will have a lot of immature embryos that you are working with, and then it's hard to make sure all the immature embryos are on the right side. And then when that happens, that immature embryo will not be able to form somatic embryos and plantlets at all. So that will be useless.
Okay, thanks. Yes, very important step. So definitely make a note of that. So I have two questions about phenolics. One is Francesca, the other is from Teal. So Teal asks, we've used PVPP in media to help with phenolics. Have you found any success with this or other additives when transforming genotypes besides RTX430?
Yeah. Using this strategy, we were able to transform 5 different varieties. Of course, you will see a bit different. You see a difference in terms of the transformation efficiency, but we are able to transform different varieties.
Okay, and then the question from Francesca, she also asked, did you use an anti-necrotic solution?
Anti-necrotic solution, is that like to avoid the formation of phenolics?
Yeah, so I'll read the full question. Were there phenolics observed? Question mark. What anti-necrotic solution did you use?
Yeah, we didn't use in our case we didn't use any kind of anti-necrotic solutions. The phenolics that you see are very minimal, and they should be gone. Like if you see sometimes, for example, for SC187, you might see a little bit more phenolics, and then it should go away with refreshing it into a new fresh medium. That should solve the problem for us. But like it depends, as I mentioned it. different genotypes might have a different response to the transformation process.
Thank you. So this next question
Sure.
is from Don Ping. Are you putting your desired gene in the same construct with baby boom and Wuschel, or are they in a separate construct?
Um, there are two different ways that you can do it. You can do it like for us, at least what we are doing is we are putting it in the same construct or in this, yeah, in the same tDNA. But there are also ways that you can use altruistic, uh, where your target gene can be in one construct, and then you will have the morphogenic genes in another construct. So when you do transformation, you can do the transformation using both agrobacteriums, especially since Wuschel will be able to initiate the formation of somatic embryos and regeneration in the neighboring cells. You can also use that strategy, but mainly we are putting it in the same construct.
Okay, thank you for that. So this next question, you had a photo where some of the plants were yellow. So we had a question from an audience member that asked, why are the shoots yellow and not green? Are they under light for shooting or maturation?
I think
Yeah, I
probably
think
this
that's what
tissues.
this is referring to. Yeah.
Yeah, yeah. At this stage, we keep them so starting the co-cultivation until the embryo maturation medium, we keep them under dark, and we move them to light when after we move them to the rooting medium. That's why we see them kind of yellowish or whitish plant plates. We also try to move them into light. We tested that earlier stage of optimizing our transformation. We moved our plantlettes into lights at a later stage on embryo maturation medium. That didn't really create a big difference in terms of the regeneration efficiency.
Thanks. So this question is from Sarah. Do you ever find plantlets with shoots that look very dry and die out during the EMM period? If so, do you know why this happens?
Um, can you repeat the question, Amy?
Do you ever find plantlets with shoots that look very dry and die out during the EMM period? I think that's the embryo maturation period.
Yeah,
If so, do you know why this happens? Um,
yeah. It's hard. It's hard. Like there might be so many things which are happening during that stage. Of course, we are using a selection agent to kill the ones which are not transformed. Uh, but yeah, there are so many components that can contribute to your plantlets. So is the question all the plantlets are dying or like some are surviving but some are dying?
I don't know, Sarah, maybe you could
Yeah.
clarify for that. Yeah, it just says plantlets with shoots that look very dry and then die. Um, I gotta say, I think this is a first for the transformation. Sarah clarified the plantlets are all dry.
Hmm. Yeah. Without knowing all the detailed informations about the medium, it's hard uh, to know what caused that. Yeah, it will be hard. There are so many components which we include in the medium. So it's hard to say. I don't know.
Yeah, yeah, interesting question. I don't think we've had that before where or I haven't seen someone have that issue with their transformation. Typically it's too much water. So this is from Umang Shar. He asked how many days okay. How many days do you change the explants into new plants or media, and how do you avoid moisture formation?
Uh, I'm
So
guessing this is on rooting medium, or is it at the early
I'm
stage?
assuming so. Yeah, it doesn't say. So, Umang Shar, if
Okay.
you can clarify. Yeah, because it just says, how many days do you change the explants into new plates or media? So, I mean, if you
Oh,
look at the
okay.
protocol, they have the number of days it should be on each of the media.
Yeah.
But yeah, I'll let you. His second question was, how do you avoid moisture formation?
Yeah. So on co-cultivation and resting medium, we keep them on average for a week. And the longest the tissues stay are on embryo maturation medium. And as I mentioned it earlier, it's recommended to refresh those tissues every week if possible, if not every other week. Regarding the condensation, sometimes that might happen. But what I will do if there is a lot of condensation is um, I'll bring, when we refresh the tissues, that should solve the problem. But it's even if the condensation happens like shorter than a week, we can move them to the laminar flow hood, let them breathe for a short period of time, and then return them into the incubator.
All right, thank you.
Sure.
So we have another question from Teal. Are individual shoots separated from the clumps unique, or do you find clones from time to time?
Both, we get both. We actually try to determine the if the plantlets are all the plantlets are independent or not. Uh, and some will be independent and some other are clonal. So it's a mix.
Yeah, we saw this with we talked about this too in the rice transformation episode that we did. When you're doing like CRISPR experiments, sometimes you have to be careful because people will pick, five plants from the same clump, and then your efficiency is super high. Well, yeah,
Yeah,
because it's you're just testing the same thing, yeah, over and over again. So, yeah,
true,
thanks for that
true.
question.
Sure. Amy, to add to that, so the reason that we are generating or moving more plantlets, like 4 plantlets from each immature embryo, is uh, to make sure, like when we move them to the rooting medium, all the plantlets might not survive on the rooting medium. So we will have extra. But at the end, especially when we are gene function study, what we are doing is finally after after the genotyping process and then after determining the transgene copy number, finally we are taking one plant from that immature embryo into the next generation.
Yeah, thank you. Yeah, thanks for that, the clarification on that. So we had kind of two questions from Mary about constructs. So her first question is, what's the biggest size of construct you have worked on? And then does your efficiency change for each construct?
Yeah. Jinchen, feel free to jump in. I think our constructs, most of our constructs that we are working on are about um, the maximum is around 35, 36. And correct me, Jinchin, if I am correct, if I am wrong. But around that stage. And we didn't see that much difference between, because the target genes that we are using, they are not that much different. From my experience, we didn't see that much difference in terms of our transformation efficiency.
Yeah, I have a comment on this question. So basically, the maximum construct we've been working on, it's about 38 in total KB. But it depends on the TDN size. So the TDN size we typically working on with less than 20 KB on TDN size. So in that regard, we haven't seen any differences on transformation frequency.
Thanks. So we got a couple of questions about the heat treatment. So I'll start with this question from Tim. Does heat-induced Cree-excision occur at 100% frequency? Are you checking by PCR that it happened in every plant that you advance?
So for the first question, not 100%, but around 75 to 85%, the excision happens on those percentage of transgenic plants that we generated. And yes, we have to genotype, do PCR on each plant to make sure the morphogenic genes are excised.
Okay, and then this question from Anthony, it just says, you do heat treatment for the baby boom Wushel construct before the regeneration step? Yes.
Yes, yes.
Okay. And then our last, so Todd weighed in on this with his experience. Thanks, Todd. But Teal asked, does the stress from heat shock affect transformation efficiency?
Not from our experience. It's just for 2 hours, and you don't see any significant effects on the tissues at that stage, and then also at a later stage.
Okay, great.
Yeah.
Yeah, so
Can
that
I
was
comment
Todd's answer as well. Oh, sorry, go ahead.
Can I comment on that part? And we have to keep the tissue in 75% relative moisturized. Otherwise, it would be dried out at 45.
Okay, thanks.
Yeah.
That's a really good note. So yeah, watch the moisture for that. So you definitely don't want it to be dry at that stage. Um, this is a question from Fizo. Um, I guess it's asking about how you came to this protocol. Yes, have you attempted to optimize the immature embryo protocol based on Aguirre 2022, such as improving promoter design or incorporating excision systems to extend its applicability beyond the 5 sorghum genotypes?
The short answer. Sorry, can you repeat the question?
Yes, have you attempted to optimize the immature embryo protocol based on Aguirre et al. 2022, such as by improving promoter design or incorporating excision systems to extend its applicability beyond the 5 sorghum genotypes? So I think at the root of that, they're
Um,
just asking what kind of optimization have you done to your protocols?
yeah, yeah. So yeah, the promoter, we didn't change the promoters. We tried, actually, we are trying to finalize a manuscript, which is a little bit different transformation strategy, which is Wushel integrated transformation strategy, which includes only Wushel only. Uh, but other than that, this transformation strategy is working well for us, especially for the genotypes that we are working with. So we didn't make that much change in terms of the protocol, which includes the baby and Wushel, but we have a different transformation strategy, which includes only WuShell that we use for genome editing. If we have a target gene that we need to edit or knock out using CRISPR-Cas9, that strategy is working well for us.
Okay, great. So this question is, have you transformed the 100M genotype?
No.
Okay. Yeah, again, the protocol lists the different genotypes that they've worked with. So if you are interested in those genotypes, you reference the protocol in the original paper. So here, another question that just came in from Fazo. Do you check for copy number?
Yes, that's one of the important steps, especially when you do gene function study. So after genotyping and identifying the transgenic planters, the next step that we do is determining transgene copy number. We use ddPCR, directly digital PCR. It works very well for us. And most of our transgenic planters, they are single copy events, around approximately 85% of the transgenic planters have a single copy insertion of the transgene.
All right, thank you.
Sure.
So this question is from Mary. Is sorghum transformation seasonal to you?
Um, like we see some difference during the winter time, summertime. Yeah. Um, we see some difference because how like it depends on how happy the plants are in the greenhouse. So I think most people have that experience.
Okay.
It's not a big difference, but still we see a small difference between seasons.
Yeah.
Yeah, I don't
Can
know what
I?
the Berkeley weather is like, if it's kind of temperate for you guys year round, or do you get a winter in California?
Yeah, like our winter, it's not as strong as the East Coast, but still, yeah, we get winter and then the temperature um, cools down during those times. Yeah, the immature embryos of the plants are not going to be as happy as they are during the summertime. Because of that, you see small differences. Jinshin, do you have?
Yeah, we did see some seasonal effects on transformation, but these differences is manageable. I would say it's about 5%-ish differences. We just start with a little bit more immature embryo to overcome the seasonal effects. This is typically what I do.
Okay, great. Thank you for that insight, because that's yeah, like 5%, that's not much. And yeah, just if you can add a couple extra to your transformation, then that should be good. Yeah, I do think this was one of the things that we discussed. If For those attending, if you weren't able to make it to our last episode, we talked about a sorghum leaf transformation. And I think that was one of the pros that was mentioned of that protocol was you don't need as much greenhouse space. So if you are in an area that maybe isn't as temperate as, yeah, California, this might be an option for you. Um, yeah, okay. So we have a few minutes left here. Um, I'm going to try to get through these last few questions here. So this is a quick one from VJ. Are your vectors available in Add No. Okay.
Okay, I will
No,
answer
yeah.
this question. The the short answer is no. So if you need that constructed, we have to sign MTA with Berkeley and also with Cultiva to get those vectors.
Okay.
Kiflin, you're welcome to chime in.
Yeah, that's true. That's correct.
Okay. So you can contact them, and then I believe their information is available in the protocols. There's the author information, so you can get in touch with someone if you want to use these vectors. So this question from Marissa, have you ever tried the Baby Boom Wuschel Sorghum Leaf Whirl transformation protocols? If you have, what were the biggest challenges? And do you feel like the media you used from immature embryo transformation could be used with leaf tissue?
Um, yeah, like our lab members, they are trying leaf-based transformation. They are still in the process of optimizing the protocol. Of course, there are differences, like different in terms of media composition between the immature embryo and leaf transformations. But it's hard for us, like we are not yet at a stage that we say we have a transformation strategy for uh, leaf. Um, so it's hard to give a suggestion at this stage.
Yeah. Yeah, and like we have the other episode. So if you are interested in the leaf transformation, you can watch that. So this is from Golem. Can I use this heat stress to other cereal crops, or on other cereal crops? So I'm assuming that's talking about the Crelox system. Um,
Probably, yeah,
but
yeah.
yeah. I'll let you guys answer that, but yeah, I just do want to remind people it's we're just covering the one protocol. So, yeah, other talking about other systems, it's kind of just I guess like not the area of expertise that we're discussing right now, but I'll let you answer the question.
Yeah, yeah, true. Different plants might respond differently to different conditions. So it's you have to taste it and see how it's working. Even the temperature of treatments, it might be some plants or some cereals might not be able to handle that high temperature. Sorghum is normally tolerant to high temperatures. So yeah, you have to taste it.
Yes, yeah. And there's other protocols, and like Teal just commented on that, it's been shown in maize. So all right. Last question here, from Francesca. Should the plantlets have a small callus base attached to them?
No, we have to remove all the tissues. When we separate the plantlets, we have to clean them as much as possible before moving them to the rooting medium.
Yeah, no, thank you for that. That's a good point because I think too it would there's potential for it to die and then become infected. Okay, yeah. Yeah, so clean those before you transfer. All right. So we are at the end of the hour. I just want to say thank you again to our speakers, for answering all of our questions. It was just like rapid fire there, and you guys did great. So thank you so much for all of the knowledge you've imparted on us today, and for sharing your expertise and your experience working with this sorghum embryo protocol. So, I just want to remind people that this has been recorded. If you are a Plant Gene member, you will get an email from me. If you are not a Plant Gene member, then you should sign up. You can go to our website. All the information is there on how to become a member. And I also want to remind people that we do have a discussion forum available. It's a great way to connect, ask further questions about sorghum, if you think of anything after this event today. And again, I just want to thank our speakers, thank everyone else for joining, and I'll see you at our next event. Great. Bye. PlantGene would like to thank the National Science Foundation for their support. To learn more, visit their website at nsf.gov.